There are these things called archaebacteria. Now, archaebacteria used to be thought of as just another form of bacteria, but DNA analysis has showed some weird stuff, and that in some ways, archaea appear to be more like eukaryotes than they are like other prokaryotes that are bacteria. So, whereas in a six kingdom sort of way of doing things, archaea are considered to be bacteria. When it comes to domains, things are divided into eukaryotes, bacteria, and archaebacteria. Archaea are very much like bacteria in format. They are prokaryotes, so all the stuff about ribosomes, DNA, so on is the same. But some of the lipids or the fats that are a part of archaebacteria are different from those in eubacteria or more common bacteria. Also, there are some differences in the cell membrane as well. The primary thing that will distinguish an archaea from eubacteria is where they live. Archaea live in extreme environments, super salty bodies of water, acidic places, places where the temperature is just ridiculous levels of heat going on. All of these are situations that would destroy regular bacteria, would melt them on spot, dissolve them on spot, whatever. But archaea, because they have these differences in their actual cell membrane and their cell wall, because they have these differences in some of the lipids within them, they're not destroyed by these extreme environments. Instead, they live in them. They live in methane environments where there's not oxygen, there's just methane. So all of these things are sort of characteristics of archaea, which are kind of like bacteria, but are more different than we thought they were. Most bacteria, they come in three basic shapes. You've got bacilli. Bacilli are shaped like rods. They look like this. They look like little rods. You see them under the microscope. This is what you see. And primarily, bacteria, eubacteria, are divided by what they look like. You've got the rod-shaped one. You've got cocci, which are shaped like spheres. They're shaped like little spheres. They get next to one another, pile on top of each other, but they're little spheres. And then you get spirilla that are corkscrew shaped bacteria. The primary difference is there. Now, remember, they all have cell walls, and that's why they have these distinctive shapes. These cell walls keep them in that particular shape. They're like hard cases around the actual bacteria, around the outside of the cell membrane of the bacteria. And that's how you get these three different shapes: the rod shaped, the sphere shaped, and the corkscrew shaped bacteria. One thing that bacteria can do, which is really, really weird, is usually you've got to have a wet environment for bacteria to propagate and grow. But they can get in this situation where they will form an endospore. And basically, what they do is they divide and they form this thick wall around the DNA and just enough cytoplasm to preserve it. They sit there as this endospore for unknown amounts of time, like years, tens of years, hundreds of years. We don't know. But these huge amounts of time, they sit there in an endospore form, because what they've done is they kind of divide, and they divide unequally, and the part that continues basically gets rid of most of its cytoplasm, most of the things necessary for life, just leaves a teeny little bit with the DNA. The thick internal wall sits there, and they are preserved in this harsh environment, this too dry environment, too cold environment, too hot environment, whatever it may be. And basically, it's like the bacteria equivalent of a seed that is able to stand up to all the difficulties around it and preserve its existence until conditions become hospitable again.